Assalamu alaikum, everyone. My name is Mustafa Ali. I am a third year medical student, and inshallah, today I'll be giving a PAL session on the protein structure, folding, and trafficking. So, in this PAL session, I'll be covering three of your MOL lectures 10, 11, and 12. I hope this session helps you guys. I know you have an upcoming midterm, you might be feeling stressed out. But hopefully this session should be enough for those three more lectures. I tried to include all of the high yield info you need to know in order to answer any of the exam questions they throw at you. I would like this session to be as interactive as possible. So please answer my questions. You could text this in the chat or unmute yourselves. If you have any questions after this session, here are my contact details, my email, and my number. Feel free to email me or message me if you have any inquiries regarding this part. Now, without further ado, let's get started. Okay, so today we'll be covering the protein structure. We'll be going through protein folding, clinical biochemistry, and then protein sorting and trafficking after that. Now, I know MOL is really annoying. I didn't like MOL in first year. It had me looking like this guy right here, right before the exam, right before the midterm. Thus, after this power session, any questions in the midterm related to these three topics, you're going to look like this when you see how easy those questions are after I'm done explaining all the high yield concepts. So inshallah, let's get started. Now, proteins are very important. They are literally related to everything that happens in our body, all of the systems, all of the mechanisms, everything. Protein has something to do with it. And it is very important. They also serve as a good food for, building muscle. If you guys are in the gym, you got to get in your protein. And it's going to be what we're going to be talking about majority of the session. But this begs the question, right? We know proteins are important, but what makes up a protein? What do you guys think? What does a protein compose of? Amino acids. Thank you very much. Now, a protein is composed of a string of amino acids that are joined together. Now, this can also be called a polypeptide. So proteins are just a bunch of amino acids connected to one another via what we're going to be talking about right now. Now, this is an amino acid, okay? Its structure might look a bit scary. It might look a bit confusing, but it is very simple. Now, an amino acid has four main groups. It has an amino group, which is right here, consists of a nitrogen and two hydrogen atoms. We have a carboxyl group, which is right here. It has a double bonded O and an OH. And we also have an R group that is present in the amino acid. Now, this R group or this side chain is very important. We're going to be talking about why it is important later on, but just keep that in mind. And we also have this hydrogen, which is by itself, that is present on the top. Now, every single amino acid has this structure. It might look a bit complicated, but every single amino acid looks like this one. Let's take alanine, for example. Now, alanine, again, has the same structure. Can anyone remind me what this group was called, or this structure is, what this part of the structure is called? Okay, thank you very much. It's called the amine group. How about this structure, which is present on the bottom? The R group, thank you very much. And how about this one on the right? Carboxylic group, thank you very much. Now, every single protein has this structure. And the R group of each of amino acids are different, okay? R groups are literally what make amino acids special in a way, because each amino acid has a different R group. Now again, R groups are what make amino acids different from one another. And we can use the R groups to classify them into different categories or different classes. Now there are four main categories or four main classes of amino acids. There are polar amino acids, non-polar amino acids, acidic amino acids, and basic amino acids. We'll be getting into each of these and looking into how their R groups look and why they are important. Now let's start with the non-polar amino acid. Before we get into the structure, can anyone tell me what non-polar means? What does it mean? When I say something's non-polar, okay, thank you very much. Hydrophobic, doesn't dissolve in water. You guys are correct. And non-polar means that it hates water, okay? It doesn't like water. And this is an example of a non-polar amino acid. Again, same structures as we talked about before. Same, it has the amine group, it has the carboxyl group, it has a hydrogen atom right here. We also have the R group right here. Now, we said that the R group is what, categorizes these amino acids into different classes, right? It's what makes amino acids different. For non-polar amino acids, they basically just have a bunch of carbon and hydrogen present, okay? Just a bunch of carbon and hydrogen present in the R group. And this is why they are non-polar, okay? And a good example would be vanillin. Do we have to know that non-polar has carbon and hydrogen? Um, in case if they do ask any questions, I do suggest that if you see an amino acid that has an R group that only has carbon and hydrogen, it is non-polar. And non-polar basically means that this is hydrophobic, okay? I hope that answered your question. Now, let's move on. Let's get into the polar amino acids and how they look. So, polar amino acids, again, does anyone know what polar means? It's basically just the opposite of non-polar. Charged, they do dissolve in water, they are hydrophilic, they love water. Thank you very much. Now, this is what happens. Polar amino acids, they love water. And what is characteristic of their R group, or what is different about their R group, is that it has a hydroxyl group that is present, or an OH, okay? Now, polar amino acids, what they have in their R group is that they have either an OH or an O, okay? And because they have either an OH or a double bonded O, this is what makes them polar. This is what makes them love water, and in other words, hydrophilic. Okay. Now, apart from this, again, we have the amine group, we have the carboxyl group, we have the hydrogen, and for the R group of a polar amino acid, it has an hydroxyl group or an OH that is present. Now, another example of a polar amino acid would be this right here, asparagine. Now, the carboxyl group does look a bit weird, so just imagine this as H. Imagine this as NH2. So this is an amine group. This is the carboxyl group. This is a hydrogen. Now, let's look into the R group. Now, in the R group, we could see that there is a double bonded O and there is an amine group. Now, whenever this is the case, okay, whenever there's a double bonded O, or another group like an amine group, it is going to be polar, okay? I said that polar amino acids either have a hydroxyl group or a double bonded O. So whenever you see either of these two, it is going to be polar, okay? Or in other words, it's going to be hydrophilic. Now let's move on to acidic amino acids and how they look. Now, for acidic amino acids, all I need you to know is that in the R group, there is a carboxyl group, okay? Same as the one that is present in nearly every single amino acid separately. That group is going to be present in the R group, right here. So there are two types of acidic amino acids. There's the glutamic acid and the aspartic acid. What you need to know is that if it's an acidic amino acid, it will have a carboxyl group that is present in the R group, okay? That is it for the acidic group. Now let's look into the basic group and how they look. Why are they different? Now, here again, it might look a bit scary. It's the same thing. We have an amine group right here, a carboxyl group, a hydrogen atom. The R group does look really long though, right? Now, we could skip through all of these carbons and hydrogens, but at the end, we could see that there's an amine group right here, right? There's an NH3. Now, this is very characteristic of a basic amino acid. They mainly have um, amine groups that are present and only amine groups that are present in the R group, okay? So again, basic amino acids, they only have amine group. They don't have a double bonded O or an OH. Just an amine group present inside of the R group will mean that it is basic. So this picture was provided by a lecturer. Um, All I need you guys to know is that these simple examples right here, methylene for the non-polar amino acids, the ones that just contain carbon and hydrogen, I just need you to remember that they are non-polar, okay? You don't need to remember the name. I just want you guys to look at the structure and be like, okay, so the R group has a bunch of carbon and hydrogen in it, so it's 100% non-polar, okay? That's all I need you guys to remember. Tryptophan is a bit complicated. Methionine is a bit complicated. Don't get into detail with those two. Just know for non-polar amino acids, they just have a bunch of carbon and a bunch of hydrogen in the R group. I don't believe so, based on the structure. I don't believe that they will ask you questions on that. I think that's way out of your scope. But just focus on how like to differentiate them from the other groups. Yes, I will. I have a slide right after this. Now, for the polar amino acids, what's characteristic of them? They have a hydroxyl group or they have a double bonded O, okay? And this is what makes them polar or what makes them love water, all right? So if they have a double bonded O like here, or they have like an OH like here or an OH like here, it's either one. It's not both of them, okay? It's not both an OH or a double bonded O. Now, if it was to be both of them that are present in the R group, it would be an acidic side chain or an acidic amino acid, okay? And if the amino acid just has an amine group that is present in the R group, then it is going to be a basic amino acid. Okay, so again, just to sum things up one more time, there are 4 classes of amino acids. They are classified based on that R group. For polar amino acids, what do they have in their R group? Do you guys remember? I think I forgot. Thank you very much. Just a bunch of carbon and a bunch of hydrogen. Okay. Wait, no. Okay, sorry guys, sorry guys. Now for polar amino acids, they either have an OH or a double bonded O. I forgot to. Polar amino acids, they contain a double bonded O or an OH. Okay. Now for non-polar amino acids, a couple of you guys answered already. What do they have? A bunch of carbon and a bunch of hydrogen. Thank you very much. And for acidic amino acids, what do they have in the R group? Thank you very much. They have both a double bonded O and a OH present. They have a carboxyl group that is present in the R group. Okay. And how about for basic amino acids? What do they have? They have an amine group that is present in the R group. Now, in the exam, just in case if they bring a structure and they ask you to say whether it's acidic, basic, polar, non-polar, just remember the R group and what makes it, a certain class. If it has a bunch of carbon, bunch of hydrogen, it's going to be a non-polar, okay? And vice versa. Okay, so let's move on. Now, histones are basically, this was a slide that was mentioned in your lecture. I decided to include it for inclusion's sake, but the reason why it was mentioned is that just one sec. But for polycystine, SH also. The person that asked the question that has a dot, all I need you to know is how like for polar amino acids, they either have a double bonded O or an OH. Cysteine is a bit more complicated. We could see that cysteine is right here. It's a bit complicated because it has an SH. They probably won't ask you about cysteine, its structure. Just focus on the most simple ones, and that has the ones that have double bonded O and the ones that have an OH. Would they give us a name of an amino acid and ask is it to identify if it's non-polar? They won't ask you for the name, okay? They'll probably show the structure and ask you to say whether it's acidic, basic, non-polar, or polar. That's probably what they're going to ask you. I feel like that would be really complicated and it would be a really hard question to answer. Okay? So you guys don't need to memorize too much. Just look at the R group and try to identify. Okay, is that good? Or can we move on? Okay, perfect. Now, for histones, it was mentioned in your slides that histones have a high content of lysine and arginine, which are basic amino acids. And they are positively charged. And you guys probably know that DNA is negatively charged. But because they're negatively charged and the histone proteins are positively charged, they will kind of bind onto the DNA and they are involved in DNA regulation and all of that. You guys probably know about it. And that's all you need to know about this. I don't think this is a relevant point in the lecture. I think it was just mentioned to kind of talk about how some proteins have basic amino acids. So let's move on. Now, essential amino acids. Can anyone tell me what an essential amino acid is, if anyone knows? Okay. Okay, I'm seeing good answers. So, an essential amino acid are amino acids that can't be synthesized by the body. We cannot make it ourselves, and they must be obtained by the diet. Now, there are 10 essential amino acids present in our body. And you can remember this by the acronym PVT TIMHOR. Now, PVT TIMHOR stands for all of these amino acids right here. Do I need you to remember every single one of these amino acids? Of course not. That would be very scary. I can tell you guys right now, I don't remember a single one of these. But what I need you guys to know is that there is one of these amino acids that is classified as a semi-essential amino acid, and that is arginine. So they could ask a silly question, which of the following is a semi-essential amino acid? It would be arginine. The reason why it's semi-essential is because it can be produced in some people whilst not in others. It's like an age thing. All you guys need to know is that arginine is semi-essential. I don't think they'll ask a question on which of the following amino acids is an essential amino acid. If they do, just remember this acronym just in case, and whatever amino acid comes to mind first related to the letter, just remember that. So I don't think they'll ask you a question on it. So let's go back to the simple definition. A string of amino acids joined together is known as a polypeptide, and proteins are made out of amino acids that are just joined together, right? But how can we join two amino acids together? That is the main question. Now, how we join two amino acids together is by joining the carboxyl group of one amino acid with the amine group of another amino acid. And we do this via a condensation reaction. A condensation reaction being a reaction that releases water. Now let's look into this picture right here. We have two amino acids, number one and number two, and the carboxyl group of this amino acid, okay, will form a bond with the amine group of this amino acid. And they will form a bond known as a peptide bond, okay? And once this peptide bond forms, it will lead to the release of water, okay? So this is what happens in a peptide bond or in a condensation reaction. Carboxyl group of one amino acid, the amine group of another amino acid come together, they release water, and now they formed a peptide bond and are connected. Now again, here is another peptide bond that forms between two amino acids, and at the same time, there will be water that is released alongside the peptide bond that forms, okay? So this is a chain of amino acids. And there's just one small concept that I wanted to explain before we move further. Now, we said that peptide bonds form between amino acids, right? The carboxyl group of one amino acid with the amine group of the other amino acid. But there is amino acids are usually connected to one another, right? And they're usually in like a chain-like shape. But there will always be one amino acid that is kind of on the edge of the chain, like this one, methylene, or this one right here. But this amino acid is not connected to another amino acid on its left, right? It's not connected to anything right here. And this one is not connected to anything right here. Third, this amino acid will have a free amine group. An amine group that is not bonded to anything. It's going to be free and lonely. And this will kind of act as the amine end of this amino acid chain, okay, of this polypeptide. Whilst this side, this amino acid will have a carboxyl group that is just left and free, and he's really lonely. And this will serve as the carboxyl end of this side, of this one right here, okay? So a polypeptide chain has an amine end and it has a carboxyl end that is just produced because the amino acid has nothing banded onto the amine group on the left side, but on the right side, there's nothing bounded onto the carboxyl group. Fair. I hope that made sense. Now we're going to be getting into the main part of the lecture, being protein structure. If you guys have any questions, feel free to ask before we move on. Mohammed, you are correct, and you are correct. I was just talking about like a very simple polypeptide chain, how it has two different ends. But we're going to be getting into how proteins kind of form and form different structures in this part of the power session, inshallah. How can I know which end is which? Usually, the amine end would be on the left of the polypeptide chain, whilst the carboxyl end is on the right. Okay? Did that make sense? Any other questions before we move on? Now, this part is going to be the main part of the lecture, protein structure. Okay, so let's move on. Now, before I get into protein structure, I want to establish a very, very important point. Now, in each level of protein structure, there are different bonds that are formed, okay? We're going to be getting into this point whilst discussing each and every one of the protein structures. So there are four levels of protein structures that you guys probably know. There's the primary structure, there's the secondary structure, there's the tertiary structure, and we also have the quaternary structure, okay? Now, let's start off with the primary structure. So the primary structure is defined as a sequence of amino acids, okay? And it is the simplest level of protein structure or protein organization. Now, the primary structure is determined by DNA, or in other words, our genetic material. This is the same picture we looked at before. A primary structure is basically just literally a chain of amino acids or a sequence of amino acids. And this sequence of amino acids is determined by DNA, and it is the most simplest organization of a protein. All right? Now let's move on to the more complicated structure, the secondary structure. Now, before I get into the structure, I would like to explain another important point before we move on. Now, in an amino acid chain or in a polypeptide chain, there are two different parts to it, or two big groups of it. We have the polypeptide backbone and we have the side chains. Now, the polypeptide backbone or the peptide backbone, it consists of amine groups, it consists of the carboxyl group of the protein, it consists of the hydrogen, it literally consists of majority of it, right? Whilst everything, it consists of everything. The polypeptide backbone has everything. But the R groups or the side chains make up the side chain of the polypeptide chain. So the side chain consists of the R groups of the amino acids, whilst the polypeptide backbone is literally everything else. The amine group, the carboxyl group, the central carbon, the hydrogen. And the reason why I'm mentioning this is because for the secondary structure, it is determined by hydrogen bonds forming in the polypeptide backbone. Okay? So yes, the R's are the side chain. Okay, so the R1, R2, R3, R4, they are considered the side chain. Whilst the polypeptide backbone consists of the amine group, the carboxyl group, the double bonded O, the H, literally everything else. And what makes up a secondary structure is the hydrogen bonds that form in the peptide backbone. Now, again, the secondary structure is determined by the hydrogen bonds that form in the peptide backbone, and it leads to the formation of alpha helixes and beta-pleated sheaths. Now, these are the two structures, or the two secondary structures. We have here, which is the alpha helix, and here is the beta-pleated sheaths. Now, for the alpha helixes, what makes their structure, it looks like a curl, right? The reason why it looks like a curl, or it's built like this, is because the hydrogen bonds that form in the alpha helix are parallel to the polypeptide backbone. Let's say the polypeptide backbone is heading in this direction. This hydrogen bond is parallel to it. In other words, this is the polypeptide backbone. This is the hydrogen bonds that form inside of it. Okay? So it is parallel. And this is why it's built like this and it looks like a curl. Whilst beta-pleated sheaths, what makes their structure is that the hydrogen bonds that form are perpendicular, they are perpendicular to the polypeptide backbone. So it's kind of like this. So, okay, to repeat one more time, the alpha helixes, the reason why they form this structure is because the hydrogen bonds that form in the peptide backbone are parallel to the peptide backbone itself. Okay? So if this is the peptide backbone, the hydrogen bonds form in this direction. But it gives off this kind of twirly structure. Whilst for beta-pleated sheaths, the hydrogen bonds that form are perpendicular to the peptide backbone. So it forms these, sheaths-like structures. We'll be getting into them more in more detail in separate slides. So for the alpha helices, again, they are formed because the bonds are parallel to the backbone. And in alpha helices, the amino acids that are present, they usually don't have bulky side chains. So amino acids like proline are never present in alpha helices. The reason for this is because proline has a really, really big R group. It's very bulky. That will lead to the structure being unstable. Another amino acid that cannot be present in alpha helices is glycine, okay? And the reason why it is not present in alpha helices is because it kind of destabilizes the alpha helix, okay? So I need you guys to remember, proline and glycine are never inside of alpha helices because they destabilize the structure. Proline is because it has a really big R group, whilst glycine is because it just destabilizes the structure. That's all I need you guys to know. Now, examples of structures or proteins that have a lot of alpha helices are keratin and myoglobin. Another important point that was mentioned in your lecture is that there was a comparison, right, between collagen and an alpha helix structure. Now, the important difference and a small little point they can examine you on is that the main difference between collagen and the alpha helix is that in collagen, there is a bunch of glycine that is present. High levels of glycine are present in the collagen, whilst in the alpha helix, there is no glycine that is present. And the reason for this is because it destabilizes the structure and it is not good. Okay? So all I need you guys to know is that proline and glycine are not present in alpha helices. The bonds or the hydrogen bonds that form are parallel to the peptide backbone, and there are high levels of glycine in collagen. Okay? If you know those 4 points or those 3 points, you'll be good to go off of the high alpha helixes. So the beta-pleated sheaths, the thing that makes them the structure is that there are hydrogen bonds that are being formed that are perpendicular to the backbone. So let's say this is the backbone. The hydrogen bonds that form are built like this. But this is why it kind of is like folded up and built like a sheet. So keratin and myoglobin, they're examples of proteins that have a lot of alpha helices. No. Okay, I'll explain it one more time. So like parallel, right? Basically means they're heading in the same direction, okay? So like, let's say if the peptide backbone is like this, okay? It's in this direction, okay? The bonds that are being formed are literally going in the same direction. They're going upwards, okay? And this is what kind of makes them coiled up and kind of like compressed because the bonds that are forming are kind of parallel. Did that make sense, Rayan? Beta-pleated sheets. Okay. So that's about it for the beta-pleated sheets. The hydrogen bonds that form are literally just perpendicular to the peptide backbone, and so it kind of folds up like clothes to sheath. Now, can anyone tell me what makes the alpha helix structure, even though I just explained it? Can anyone tell me why it is built like this, why it has this structure? What is the most important thing to know? Thank you very much. There are hydrogen bonds that form that are parallel to the peptide backbone. And how about the beta-pleated sheaths? Okay. They are perpendicular. Thank you very much. And what two amino acids are not present in alpha helices? Okay, that's good. Proline and glycine. Okay, thank you very much. And those are the main points you need to know. And also, um, inside of collagen, there is a lot of glycine that is present. Just a silly point they could examine you on. Now, let's move on to supersecondary structures. Now, it's in the name. Supersecondary basically means a structure that has a bunch of secondary structures that are present. They are known as motifs, okay? So motifs are basically just a combination of secondary structures. An example could be an alpha helix here and an alpha helix here, or beta pleated sheath and another alpha helix and another beta pleated sheath. And this is basically what was provided. This is all you need to know, okay? Now, domains are basically the combination of supersecondary structures. Or in other words, they are the combination of motifs. And they are functional and three-dimensional structural units of a polypeptide. And a large protein can have a lot of domains or multiple domains. Each domain is structurally and functionally independent of other domains. A good example could be like, let's say if you have a channel and then there is a binding site for that channel, the ligand that binds onto it and causes the channel to open methylene. This is called a domain. This site is called a domain because it has its own function and it has its own structure. Okay? And its domain is basically just a combination of supersecondary structures. So I hope the secondary structure made sense. Now we'll be moving on to the tertiary structure and seeing how that looks. Now, for the tertiary structure, again, we're on this side. We talked about previously that for the secondary structure, it's basically, well, one sec, can you explain motifs again? Okay, all I need you to know is that motifs are just literally a combination of secondary structures. There could be two alpha helixes or two beta pleated sheaths, or one alpha helix, one beta pleated sheath. So, simple terms, it's just a combination of secondary structures. Okay? Did that make sense? Okay. So, I hope the domain made sense too. Let's move on to this one more time. Rayan, so motifs are domains. No, no. So domains are literally a combination of motifs, okay? So like there could be two motifs that form a domain, three motifs that form a domain. A domain itself is just a combination of supersecondary structures. Thank you very much, no, you're correct. Motifs are not functional, whilst domains are functional, okay? So yes, you're correct. Domains are tertiary structures, okay? That is also something you guys should know. I was going to mention it in the tertiary structure section, but thank you very much. Hey, can we move on? Or do you guys have any other questions? Yes. Side chains are the R groups. Yes. Okay. So can we move on? All right. So this picture again. We talked about in the secondary structure, there are hydrogen bonds that form in the what? The peptide backbone, right? We talked about that. We talked about the alpha helixes, the beta pleated sheaths. We talked about how the hydrogen bonds form in the peptide backbone. Okay? Easy enough. Now for the tertiary structure, what is different here is that now there will be bonds that form in between the side chain, okay? Or the side chains. So now bonds will be forming in between the R groups of these amino acids. And this is what gives the tertiary structure its structure. So let's put this into words. Now, the tertiary structure is basically the formation of a 3D structure, which is folded. And this structure forms because or due to the bonds between the R groups or the side chains. Now, the types of bonds that form are hydrophobic interactions, ionic bonds, hydrogen bonds, and disulfide bonds. Now, this picture was provided by your lecturer. You could see here that hydrophobic interactions kind of cause the peptides to kind of get away from each other. The hydrogen bonds can form between hydrogen and oxygen present on different amino acids. There are ionic bonds that form, which are bonds that are between negatively charged and positively charged ions. And we also have disulfide bonds, which are really, really strong. They mainly form between cysteine residues, so that could be something, a little cheeky question they could ask. But all you guys need to know is that in the tertiary structure, there are these four interactions that happen. And where do they happen, or between which groups do they happen in? In between the R groups, okay? And this is what gives them that folded and that functional structure. And I hope that made sense. Now let's move on to the quaternary structure, which is kind of the final puzzle or the final piece or the final functional protein. Now, quaternary structure is basically defined as different polypeptide chains coming together to form the final protein. Two polypeptide chains are called like a dimer, whilst three polypeptide chains together form a trimer, whilst four polypeptide chains would form a tetramer, okay? And quaternary structure is just the finalized protein, all right? An example of a quaternary structure would be hemoglobin. You can see it has a chain here, it has another chain here, it has another chain here, and has another chain here. So this is an example of a quaternary structure. How about myoglobin though? Does anyone know if myoglobin is a tertiary structure too, or is a quaternary structure? Does anyone remember? Thank you very much, Miss. Now, myoglobin is a tertiary structure, and the reason for this is because it has a single polypeptide. It doesn't have multiple polypeptides that kind of come together. Okay, so these are the four levels of protein structure. We have the primary structure, which is the most simplest. Okay, it's literally just a sequence of amino acids. We have the secondary structure, which is just made out of a bunch of alpha helixes, beta sheaths. The reason why they do form is because there are hydrogen bonds that form in the polypeptide backbone. Okay? Now, for the tertiary structure, which is the next step, there are bonds that form now between the R groups. Okay? So there are bonds like the ionic bonds, the hydrogen bonds, hydrophobic interactions, and disulfide bonds that form in between the R groups of these amino acids. And then we have the final puzzle, or we have the final product, sorry, being the quaternary structure. Now, the quaternary structure is literally defined as multiple polypeptides kind of coming together to form that final product and become very functional. Okay? And those are the four functions or the four protein structures that are present in our body. Does anyone have any questions before we move on? Oh, I do believe methionine can form a disulfide bond, if I'm not mistaken. I do believe so. Okay, tertiary structure isn't clear. What do you want me to explain about it? So, okay, to put it in simple terms, right? The tertiary structure is the structure that forms when there are interactions or there are bonds that form in the side chain of the amino acid. So what do I mean by this? This R group can form a bond with this R group. This R group can form a bond with this R group. This R group can form a bond with this R group. The interactions between the side chain of the whole polypeptide is what forms the tertiary structure. And we have four interactions that can happen: the ionic bonds, the hydrogen bonds, the hydrophobic interactions, and the disulfide bonds. So this is what kind of gives the tertiary structure its folded, look. Okay? Did that make sense, Abdullah? Or should I repeat one more time? Okay. Now, to put this in simple terms, a dimer, a D in the name, so it's just two polypeptide chains. A trimer has three polypeptide chains. A tetramer has four polypeptide chains. Okay? Did that make sense? No? And again, quaternary structure is literally just the final product. It's multiple polypeptides coming together and forming that final product. What about collagen triple helix? Now, okay, this was a point that I mentioned on this slide right here. Now, all I want you to know about collagen or the triple helix structure of collagen is that inside of collagen, there is a high level of glycine. Okay, there is a bunch of glycine that is present. And because it is present, this is what makes it different from an alpha helix. Okay, alpha helixes, they have no glycine and they have no proline, right? We talked about how they kind of instabilized or destabilized the structure. Whilst in a collagen, they have a bunch of glycine. That's the only difference I want you to remember, and the only thing they could kind of examine you on in case. Did that make sense? All right. Is there any other questions before we move on? Going to be getting into the next lecture right now. No questions? Okay. So let's move on. Now, protein folding. Inshallah, this part is easier. Now, a folded protein is a functional protein. Folding is very important for giving the protein, or making it functional so it can be released and do its role. Now, a misfolded protein, vice versa, a misfolded protein can lead to loss of function or even disease. Now, there are these proteins called chaperones. Now, chaperones are responsible for protein folding and they prevent misfolding too. An example of a chaperone would be HSP70, which is a chaperone that is present inside of cells. It kind of folds them up, make them neat and tidy, and kind of allows them to go do their function and become functional. Now, chaperones, they use energy to fold proteins. And this image kind of displays it. Now, what a chaperone does is that it allows an unfolded protein to go inside of it. It kind of closes it, and using ATP, which is energy, it kind of folds up the protein and allows it to become functional and then releases it into the world. Okay? So that's what a chaperone does. Now, there's this other concept called melting temperature. And what melting temperature essentially is the temperature at where 50% of a protein is unfolded. Okay? And most proteins, melting temperature is literally around 40 to 60 degrees Celsius. And the higher the melting temperature of a protein, the more stable that protein is. It makes sense, right? The more heat that is required to misfold a protein, the more stable it is. That's all I want you to know about the melting temperature. Just know the definition, and that's about it. Now, for denaturation, can anyone tell me what denaturation means? If any of you guys remember or not? Okay. Okay. Now, okay, most of your answers are correct. Most of your answers are correct. Now, denaturation, in terms of definition, it is the loss of secondary and tertiary structure without breaking peptide bonds. Now, all of your answers are correct. In simple terms, it's when the protein loses its structure, and it can lose its structure due to a lot of different things. It's when the protein becomes unfolded, it loses its structure, and it could be caused by things like heat, a very high temperature, a very low temperature. It could be caused by acidic pH, a basic pH. It could be caused by a lot of different things. What these essentially do is that they lead to the protein structure of the protein being lost. Okay? And it's usually irreversible. You can't reverse the denaturation of a protein. Once it's gone, it's gone. And that's about it. Is that the same thing that happened when the H bond? Yes, yes. That's what happened. You're right, Mohammed. So denaturation basically is just the loss of structure of a protein, and it can happen due to heat, acids, bases, and it's when the secondary and tertiary structure are lost without breaking the peptide bonds though. The peptide bonds are still kept in between the proteins or in between the amino acids. Now let's move on to protein misfolding. We talked about protein folding and why that is important. The proteins that were responsible for protein folding were the chaperones. Now let's move on to protein misfolding. Now, what's happened here is that usually in our body, we usually break down these misfolded proteins, okay? These misfolded proteins, they're not good. And if they are not broken down by our body, they can aggregate. What do I mean by aggregate? Now, aggregating basically means when proteins kind of stick onto one another and they kind of form these big, large complexes. Now, aggregated proteins are toxic. Why? Because they can become really big. And what can they lead to? Now, you can think about aggregated proteins like kind of like cement and bricks, kind of sticks the bricks together, right? Aggregated proteins or misfolded proteins, sorry, are sticky in nature. They stick onto one another and they become really big. And what can they lead to? Now, they can lead to disease. And this is very bad. But what can cause protein misfolding in general? What can literally lead to these proteins becoming misfolded and potentially lead to aggregating proteins and then lead to disease? Now, things like DNA mutations can lead to protein misfolding. There being changes in the genetic material can lead to this. Things like stresses, like high temperatures, low temperatures, high pH, low pHs. Environmental factors like toxins and pesticides can lead to protein misfolding. And even age. Now, age is just basically due to the chaperone proteins kind of getting tired in an older person. It's been working a long life, right? So it kind of, slacks off, and this causes protein misfolding slightly. But yeah. So how do we deal with misfolded proteins? Now, some of these factors, right? Stresses, methylene, or environmental factors, we are probably exposed to like toxins or pesticides or even metals throughout our daily lives, right? We're exposed to some of these factors. Age is a normal thing, so stresses like high temperatures, low temperatures, like we live in Riyadh, but it's really hot. Now, all of these things can lead to protein misfolding, and protein misfolding can lead to disease. So why don't we all have diseases? Allah yusuf. Best. How does our body really deal with these misfolded proteins? What can it do? Does anyone want to answer? How does our body try to, deal with these misfolded proteins and get rid of them, maybe? Okay, okay, break them down. I'm seeing some good answers. Now, can anyone tell me what does this? I see someone wrote a good answer. Now, you guys are correct. They are broken down. And what they are broken down by is the ubiquitin proteasome system. Okay, the ubiquitin proteasome system. Now, this is what the system is. What essentially happens is that there is an unfolded protein present, and this unfolded protein is marked with something called ubiquitin. Okay, once this protein or this misfolded protein is marked with the ubiquitin, it kind of causes this proteasome to kind of wake up and become alert. And this proteasome, it will eat up this misfolded protein and ubiquitin. It will eat it up. And once it's eaten it up, it will lead to the protein being broken down, being shredded up. And this is what happens with the ubiquitin proteasome system. It sees that there is a unfolded protein or a misfolded protein. It puts ubiquitin onto it. Once ubiquitin is on it, this proteasome will come to this ubiquitinated protein and it'll kind of eat it up, break it down, and it spits it out. Now it's not misfolded anymore, okay? And it can't cause disease. So this is what the ubiquitin proteasome system does. Does it make sense? Exactly. Thank you very much, Mohammed. So, but I have another question for you guys. Let's say that the ubiquitin proteasome system is trying to break down all of the misfolded proteins inside of our body. It's getting to work. It's working really hard. But there is just too many misfolded proteins that are present. There's too many. What will happen? And it's working as hard as it can, but it cannot work enough. To be simple, what happens is disease can occur. Okay? And this brings us to our next topic being clinical biochemistry. We'll be discussing a couple of cases, how DNA mutation can potentially lead to misfolded proteins, which could lead to diseases, or how protein aggregation can kind of also lead to diseases. And you'll be getting into them with a couple examples that we'll mention in your lecture. Before we move on, does anyone have any questions they would like to ask Or are we all good? Any questions? Okay. Yes, there will be practice questions at the end. There's a quiz that is in the QR. But user, what don't you understand? What do you want me to go over? Okay, so what essentially happens, right, with the proteasome? Simple terms, there's a misfolded protein. What the system does is that it puts these green things right here, called ubiquitin. It puts it onto the protein. Once it puts it onto the proteasome is signaled to kind of come and eat it up. Once it's eaten it up, it then releases it and kind of goes about its day. But that's what happens with the ubiquitin proteasome system. Okay? Does it make sense? Okay. Let's move on to clinical biochemistry. Now, again, the primary structure of a protein is determined by DNA, the genetic material. But what if we have a mutation? What's going to happen? Let's find out. Now, let's get into cystic fibrosis and what happens there. So, in a normal person, right, we have a CFTR channel, okay? Now, this CFTR channel, what it does is that it essentially just gets chloride out of the cell, okay? And what this will do is that it will kind of hydrate the mucus that is present on top of the cell or outside of the cell. It makes sure it's thin, tidy, and not sticky. That's all it does, right? So it releases chloride to make the mucus outside of the cell more thin and less sticky, okay? This isn't a normal person, and this is what the CFTR protein does. Now, in a person with cystic fibrosis, they have a mutation. And this mutation occurs in a place called delta F508. Once this mutation occurs, it will lead to the deletion of an amino acid. Okay? And this amino acid is phenylalanine. Okay? Now, because it leads to this amino acid being deleted, or it is not going to be present in the protein structure anymore, wouldn't this mean, right, that the primary structure would be affected? We said that the primary structure is literally the sequence of amino acids or amino acids joined together in a chain, right? That's what we said. If there is an amino acid that is deleted, in this case it's phenylalanine, what will happen is that one of these amino acids will be cut off or removed. This will lead to the primary structure being different now. Now, because the primary structure is different, this will lead to the secondary structure being different too, right? Because it's basically just the primary structure put together. And because the secondary structure is affected, the tertiary structure will also be affected. Because the tertiary structure is affected, what will lead to the quaternary structure? What do you guys think? It will be abnormal. The domino effect, exactly. For because bad stuff, thank you very much. For because of this deletion or because of this mutation, phenylalanine is not present. Because phenylalanine is not present, it leads to the CFTR channel becoming weird, becoming abnormal. And because it's abnormal, it can't do its function, which is to get chloride outside of the cell. And because it can't do this, it leads to there just being a bunch of sticky mucus that is outside. That kind of leads to the person getting multiple infections, leads to them having growth issues. And this is what happens in cystic fibrosis. What I want you guys to remember is that there is a mutation that happens in this site called delta F508. It leads to there being a deletion of an amino acid, which is phenylalanine, and it leads to the CFTR channel being abnormal and not doing its function. If you guys can remember those 3 points for cystic fibrosis, I think you're set to go. Exactly. They do affect every single organ. They affect like the pancreas, the lungs, could affect the GI tract too. So it does affect a lot of different areas. But I just want you guys to remember these 3 points. Okay? So I hope that made sense. Let's move on to sickle cell anemia and what happens there. Now, we're looking at hemoglobin, okay? We looked at this picture before, right? Can anyone remind me what the structure of hemoglobin was, or what structure it has? Does anyone remember? As a quaternary structure. Thank you very much. As a quaternary structure. Why? It's because it has 4 polypeptide chains that are just joined together. Now, hemoglobin is the star of the show when it comes to sickle cell anemia. What happens essentially is that there is a mutation that happens. There is a mutation. The mutation that happens in sickle cell anemia is called a point mutation. Or in other words, a single nucleotide is changed. You could see, right, there's an A here instead of a T, okay? And this is what happens in sickle cell anemia. There is a single nucleotide that changes. And because of this, or in other words, because of this point mutation, what happens is that there will be a substitution of amino acids. There will be a substitution of glutamate with valine. And because of this substitution, glutamate, in nature, it is hydrophilic. Hydrophilic basically meaning that it loves water, whilst valine is hydrophobic, meaning that it hates water. Because there is a substitution between these two amino acids, it will lead to this sickle cell anemia. And what happens in sickle cell anemia is that the red blood cells kind of become sickle in shape. They become crescent in shape like this. And the reason why this happens is because of the hydrophobic group of valine that is present in this hemoglobin. Because of this substitution with glutamate, which is hydrophilic, and valine, which is hydrophobic, this hydrophobic group, this hydrophobic amino acid, because it hates water, it'll try to like curl up and hide itself. So because it does this, it would lead to like the sickle cell shape inside of these RBCs. And all of this happens just because of the single point mutation, which led to the substitution of glutamate with valine. Okay? If you guys can remember these 2 points, that in sickle cell anemia, there is a point mutation. Glutamate, which is hydrophilic, is substituted with valine, which is hydrophobic. You guys will be good to go with sickle cell anemia. Okay? Yes, you are right, no. It is a missense point mutation. Now, again, because there was a substitution in amino acid, now it is not being deleted. It's literally just being changed with another amino acid. And we said the primary structure is basically the sequence of amino acids, right? If one of the amino acids is switched with another, it will lead to the primary structure being affected, right? Now, because of the primary structure being affected, the secondary structure would be affected. Because the secondary structure is affected, it leads to the tertiary structure being affected. Because the tertiary structure is being affected, what will happen to the quaternary structure, do you think? It will be affected. And this is all due to that single point mutation that's happened between valine, which led to the substitution of glutamate with valine. Did that make sense, guys? You can say glutamic acid, but glutamate is usually mentioned. Okay? Did it make sense? All right. So let's move on to Huntington disease. Now, Huntington disease is basically a neurodegenerative disorder. What do I mean by this? It's a disease that kind of eats up on the brain and breaks it down. And it leads to, this is a normal brain right here. It leads to the brain being, smaller, having a bunch of holes in it. And this is what happens in Huntington disease. Now, what's more important is what causes it. Now, the most important thing you guys need to know about Huntington disease is that there is a polyglutamine repeat expansion. What do I mean by this? Now, in a normal Huntington protein, that is present inside of our body, there is a lot of glutamine that is present inside of it, okay? Normally, there is a lot of glutamine that is present inside of it. But with Huntington disease, when there is a mutation, this mutation leads to there being even more glutamine DNA, okay? Or even more DNA that codes for glutamine. What this will lead to is that there will be so much more glutamine that is present in the protein. And this will lead to the protein then becoming abnormal because there is a bunch of glutamine that is present inside of it. And because the protein or the Huntington protein becomes abnormal, they will then aggregate onto one another. Okay? They'll stick onto one another inside of neurons. And we said, right, when proteins aggregate and they stick onto one another, they become toxic. And these aggregates will be present in neurons. So they'll target the brain, the central nervous system. And this is what happens in Huntington disease. All I need you guys to remember is that there is a polyglutamine repeat expansion that happens here. It leads to there being an abnormal Huntington protein. They stick onto one another, or in other terms, aggregate inside of neurons, which leads to the neurons dying. Okay? Did that make sense? Okay. I'll explain it one more time. All right. So Huntington disease is basically a neurodegenerative disease. It breaks down the brain. What I need you guys to know is that in Huntington disease, there is a polyglutamine repeat expansion. To put in simple terms, what happens in Huntington disease is that there is more glutamine that is present in the protein. Okay, and the reason why this happens is because there is more codons that code for glutamine, sorry, that are incorporated into the Huntington protein. So in a normal person, there would be like let's say there would be like five glutamine, there's five glutamine inside of a normal Huntington protein. In a person with Huntington disease, they would have like 30. Okay, that because there is more glutamine that is present in a person that has Huntington disease or inside of the Huntington protein that is mutated, it will lead to the protein becoming abnormal. When it becomes abnormal, it can then aggregate in neurons. Once they aggregate in neurons, it can then kill them. Okay? But all I need you to know or remember is that there's a polyglutamine repeat expansion. It leads to there being more glutamine inside of the Huntington protein, which leads to it sticking onto one another and aggregating, which leads to it killing the neurons. Did that make sense? Okay, so let's move on. Now, amyloid diseases, okay? So, amyloid diseases are caused by protein aggregation, which are made up of amyloids, all right? And they become really toxic. We said when proteins aggregate, it's a big no-no. Becomes very toxic to the cell and can kill it. Now, in amyloid diseases, these amyloid aggregates can accumulate in different organs and lead to dysfunction in these different organs. And we'll be looking at three amyloid diseases. We'll be looking into Alzheimer's disease, Parkinson's disease, and then cardiac amyloidosis. So let's get into the Alzheimer's disease first. Now, Alzheimer's disease is a very common condition that happens with age. It's a very heartbreaking disease. The elderly start to lose their memory. They start to be confused all the time. They don't know how to do familiar tasks. And it's a very heartbreaking disease. But what happens in Alzheimer's disease, that I need you guys to remember, is that there is this enzyme called secretase, okay? Now, in Alzheimer's disease, this secretase is abnormal, okay? And what this secretase enzyme is supposed to do in a normal person is that it's supposed to break down this protein called the amyloid precursor protein, or the APP, okay? So the secretase is supposed to break it down, all right? Now, because the secretase in Alzheimer's disease is abnormal, it will cut down or break down the APP abnormally, all right? Once it breaks it down abnormally, it will lead to this fragment being released called what? The amyloid beta protein. And now, because this amyloid beta protein is released, it's not supposed to be released. It's only released because of this abnormal secretase enzyme. It loves to stick, okay? And it loves to stick to different amyloid precursor protein, all right? Now, because this amyloid precursor protein is released, it will kind of stick onto other amyloid beta proteins. Sorry, wait. Because the amyloid beta protein loves to stick onto other amyloid beta proteins, they'll kind of stick on top of each other. And as you can see here, we just have a bunch of aggregated amyloid beta protein. So this is what happens. And we said that aggregated proteins are really toxic to the cell. And this could lead to a lot of issues. So again, to repeat one more time, what happens is that there's this enzyme called secretase. In Alzheimer's disease, this secretase kind of breaks down this APP abnormally. Because it breaks it down abnormally, it kind of leads to this amyloid beta protein right here being released. This amyloid beta protein loves to stick to other amyloid beta proteins, and it would form these big aggregates right here, or these big chunks of protein, and they become toxic to the cell, and they form things like amyloid plaques. Okay, did that make sense? All right. So this is one part of the disease. Now, something else that happens in Alzheimer's disease is that there are these tau proteins. Now, what these tau proteins do in a normal person is that they kind of are responsible for like the microtubules inside of the cell. They're important for structure. Anyhow, tau proteins in Alzheimer's disease, they get hyperphosphorylated. Okay? They get hyperphosphorylated. Because they get hyperphosphorylated, they become really sticky. Okay? They become really sticky. Once they become really sticky, they form these neurofibrillary tangles. Okay? And they form these neurofibrillary tangles, which are present inside of the cell. Okay. Um, did the tau protein make sense? Before I'll explain them both together one more time. But did the amyloid, all I need you to know from the slide is that the tau protein gets hyperphosphorylated. Because it gets hyperphosphorylated, it gets really sticky. They stick onto one another, or in other words, they aggregate, forming these neurofibrillary tangles. Okay, to repeat this one more time for the amyloid plagues, what's happened is that there is this, yes, exactly. Thank you, Mohammed. For the Alzheimer's disease, what happens is that there is the secretase enzyme, right? Which is supposed to cleave or break down this APP protein. Now, in a person with Alzheimer's disease, this secretase enzyme is abnormal. It's weird. And because it's abnormal, it will cut this APP in a weird way. All right? And because it cuts it in a weird way, it kind of leads to this fragment called the amyloid beta protein being released. Okay? And now, because this amyloid beta protein is being released, one's being released from the left, one's being released from the right, one's being released from the center. Something about these amyloid beta proteins is that they love to stick onto one another. And because they love to stick onto one another, they would kind of form these big protein structures right here. They would aggregate and they'll become toxic to the cell. And they'll form something called amyloid plaques. Okay? So all you need to know, abnormal secretase cuts off APP in a weird way. Amyloid beta protein is produced, very sticky, sticks onto other amyloid beta proteins, then it leads to disease. Okay, Mohammed, I understand what you're asking, and it is a very good question, but it is not, required to know the difference between these two secretases in your exam. You just need to know that it is abnormal and it cuts this APP abnormally. Okay? So it's not too relevant for your like exam material. Okay. So I hope that made sense. Again, what happens in Alzheimer's disease is that there's two things. Can anyone remind me of the two things that happen in Alzheimer's disease? If anyone knows. What happens in Alzheimer's disease? Can anyone tell me like what causes the disease? Okay. Okay. So thank you very much. It's the tau neurofibrillary tangles and it's the amyloid plaques. The amyloid plaques form because of the abnormal cleavage by the secretase, which leads to this amyloid beta protein being released, which leads to it sticking onto other amyloid beta proteins, which leads to these big aggregates or these amyloid plaques. Now, for the tau tangles or the neurofibrillary tangles, what's happened is that there are these tau proteins which are phosphorylated or hyperphosphorylated. They become really sticky, they stick onto one another, and they lead to these neurofibrillary tangles forming. Yes, you're right, Mohammed. You're right, Mohammed. So this is what happens. Both of these contribute to Alzheimer's disease. Does anyone have any questions before we move on to the next disease? Does anyone have any questions? Okay, all good. Now let's move on to Parkinson's disease. So Parkinson's disease is another heartbreaking disease. One second. Is Alzheimer's disease just a big brain clog? You could kind of think of it like that. It's caused by like these protein aggregates, right? The neurofibrillary tangles and the amyloid plaques, which kind of are present inside of the neurons, which kill them. So they kind of like break down the brain, okay? If that makes sense. Because they're really toxic to the neurons. Okay. So are you talking about this slide, Lamar? So this slide, I'm just basically reminding you guys that there are two things that happen in Alzheimer's disease. There is the amyloid plaques that form, and we talked about why they form, and there are the neurofibrillary tangles that form. Okay? Okay, so the neurofibrillary tangles, it forms because there's this protein called tau. There's this tau protein, right? This tau protein gets hyperphosphorylated. Because it gets hyperphosphorylated, it gets really sticky. And because it gets really sticky, it'll kind of stick onto one another, forming these neurofibrillary tangles or these like big chunks of tau protein. Did that make sense, Mohammed? Um, did that make sense, or should I repeat one more time? All good? Okay, okay. Now, that's a very good question, Lamar. 5 plus 5 is 10, right? Yeah, it's 10. I'm not good with maths. But let's move on to the another disease, Parkinson's disease. Now, Parkinson's disease is another neurodegenerative disorder. They usually have like a tremor. They usually have dizziness. They forget stuff too. And it's another heartbreaking disease. Now, what I want you guys to know about Parkinson's disease is that there is misfolding of a protein. And this misfolding of a protein is the alpha synuclein. Okay? So the alpha synuclein becomes misfolded. They aggregate onto one another and they form these Lewy bodies, which are present inside of neurons. So all I want you guys to know is that there is misfolding and aggregation of alpha synuclein. Because of these, this alpha synuclein or the aggregates that it forms form these Lewy bodies. Okay, these Lewy bodies are present inside of neurons, and they become toxic to the neurons, and they kill them. All right. If you guys can know these 2 points, alpha synuclein, they form Lewy bodies inside of neurons, toxic to the neurons. GG's. All right. Did that make sense? Okay. So let's move on to cardiac amyloidosis. Now, this condition, do I need you to remember this whole slide? Wallahi, I don't. Okay? All I need you to know is that there is an accumulation of a protein called transthyretin. And because of this accumulation, it kind of sticks onto one another, forming these aggregates. It can kind of go to the heart and need to restrict the cardiomyopathy, or it could go to like the periphery, leading to like peripheral neuropathy. Do I need you to remember both of these? No. I just need you to know accumulation of transthyretin protein, goes to the heart, becomes toxic there, goes to different other places, becomes toxic there. Just remember the transthyretin protein and that it aggregates and can lead to cardiac amyloidosis. Okay? That's all I need you guys to know. I'm not perfectly clear about Parkinson's. All I need you to know about Parkinson's, Ya Muhammad, is that there is misfolding and aggregation of alpha synuclein. Okay? Because it kind of aggregates and misfold, it forms these Lewy bodies, which are present inside of the neurons, and they become really toxic to the neuron and kind of kill it. Did that make sense? That's all I need you to know. All the symptoms I talked about before, like this stuff, wallahi, you don't need to remember these. Can we move on? Exactly. Exactly. Yeah, you're correct. Now let's move on. So the last part of this lecture is prion disease. Now, a prion is an infectious protein. What do I mean by this? Now, let's say this is a prion right here, right? A prion is a misfolded protein. Let's say this is a normal protein right here. So here is the prion, here is the normal protein. What a prion does, right, is that it can touch the normal protein and wow, it makes it misfolded. For a prion is an infectious protein. Why? It's because it can kind of spread its misfoldedness to different proteins. But if the prion itself is misfolded, if it touches another protein, it becomes misfolded. And that's what it can do. For prions are very scary, right? Trade one prion inside of your body can cause all of the other proteins inside of your body to become misfolded. It's very scary, right? Now, prion disease mainly targets the brain. It can lead to neuronal damage. The umbrella term that you guys should know for prion disease is transmissible spongiform encephalopathy. It's a very long name, right? Let's break it down. Transmissible, basically meaning that it is infectious. It could transmit from one person or one protein to the next. Spongiform, basically meaning it makes the brain look like a sponge. It kind of puts a bunch of holes in the brain, hence looking like a sponge. Encephalopathy, basically meaning that it affects the brain. Okay? And you guys should remember this term in case they ask a question. Now, prion disease in cows is known as mad cow disease, okay? But the more technical term is BSE, or bovine spongiform encephalopathy, okay? Bovine spongiform encephalopathy. In sheep, the prion disease is called scrapie. All I want you guys to know is that these definitions for different, organisms, for like cows, it would be BSE. For sheep, it would be scrapie. All I need you guys to know is remember the names in case they ask a silly question. I do believe it was on your slides, but that's why I'm just mentioning it just in case. And there's this disease, of course, called Creutzfeldt-Jakob disease. Do you guys need to know everything about it? No. Just need to know that this is prion disease in humans. It causes the brain to shrink. Humans can get it through like cadavers, so be careful in the lab, wear your gloves. They could get it through like eating infected cows, infected sheep, and it basically eats up on the brain and kind of shrinks it. So just be familiar with these names, BSE, Scrapie, and be like familiar with transmissible spongiform encephalopathy in case if they ask a question. And that is about it for this lecture. Does anyone have any questions before we move on to the last topic? No question is a bad question. So if you want to ask anything, ask it now. Now, the names of the prion diseases. For cows, you could call it mad cow disease, or you could call it BSE, or bovine spongiform encephalopathy. For sheep, it would be called scrapie. And for like the human thing, I don't think you need to remember this, but Creutzfeldt-Jakob disease, or whatever it's called. You don't need to remember that though. Now for TSE, it stands for transmissible spongiform encephalopathy. Transmissible, infectious. It can transmit from one protein to the next, right? Said that this is a prion, this is a known protein, touches it, oh, it's misfolded. Spongiform basically makes the brain look like a sponge, puts a bunch of holes in it. Encephalopathy, basically meaning it infects the brain, and that's about it. Okay, I just explained it for the fun of it, by the way. Now that's about it. Do you guys have any questions before we move on? The E stands for encephalopathy, which basically means that it infects the brain, or that if it affects the brain. That's all it means. Okay. Is that about it? Does anyone have any questions before we move on? Okay. So now we're at the last point. So in protein processing, right, you guys have probably studied about transcription, translation, all of that stuff. Transcription happens in the nucleus. And once transcription has happened, it leads to an mRNA being produced. Now, after transcription of the DNA that is in the nucleus occurs, there are two different pathways that the mRNA can go through. Okay? It can go through the secretory pathway, and it can also go through the non-secretory pathway. Now, if it goes through the secretory pathway, it can lead, or if it goes through the secretory pathway, what will happen to the mRNA is that it will get translated on the RER, or on the rough endoplasmic reticulum. If the mRNA goes through the non-secretory pathway, it will get translated by a cytosolic free ribosome, or a ribosome that is free in the cytoplasm. Okay? So if it goes through the secretory pathway, the mRNA gets translated in the RER. If it goes through the non-secretory pathway, it gets translated on a free ribosome in the cytoplasm. Okay? Does that make sense? Okay, so let's move on. Now, for the non-secretory pathway, again, what happens is that the transcription happens, mRNA goes out. The mRNA would get translated by a ribosome just in the cytoplasm, free as a ball, and it gets translated and the protein is produced. Now, the protein that is produced, if it goes through this non-secretory pathway and it is produced, it will go to 4 different sites. It will go to the cytoplasm, it will go to the nucleus, it will go to the mitochondria, or go to the peroxisomes. The proteins that undergo the non-secretory pathway, or the mRNA that undergoes the non-secretory pathway, they will go to these 4 sites, okay? Being the cytoplasm, the nucleus, the mitochondria, or the peroxisomes. They are destined for these sites. Simple enough, right? Now, for the secretory pathway, what basically happens is that there are proteins. And these proteins, instead of being translated on a free ribosome, they are going to be translated on the RER, or in other words, in this area right here. Okay? And the proteins, or the mRNAs that are translated in the rough endoplasmic reticulum or around the area, they can go to these 5 different sites. They could stay in the ER, they could go to the Golgi, they could go to the lysosome, they could go to the plasma membrane, they could even go outside of the cell and be released. Okay? So in the secretory pathway, the mRNA gets translated in the RER or around the RER, and it can go to these 5 different sites, being the staying in the ER, going to the Golgi, can go to the lysosomes, it could go to the plasma membrane, and it could leave the cell and go to the outside. But I have a question for you guys. How does the cell determine whether a mRNA should enter the secretory pathway or the non-secretory pathway? How can the cell decide this? How does the cell know? Can anyone tell me? No, I see a lot of good answers. The answer is signals. Okay? Thank you very much. Okay, thank you very much. Now, the answer is the N-terminal signal peptide. Okay? The N-terminal signal peptide. Now, if the protein or the mRNA has the N-terminal signal peptide, it will go through the secretory pathway. But if it doesn't have the N-terminal signal peptide, it will go through the non-secretory pathway. Okay? So if the N-terminal signal is present, secretory pathway. If it's not present, the non-secretory pathway. Simple enough, right? Now, this picture was present in your lecture. It looks very confusing, right? Let me try to simplify it as much as I can. So, what happens in the secretory pathway, we said, is that there is an mRNA which goes to the RER and gets translated there, right? Before it goes into the RER, it attaches to the ribosome before it goes into the RER, okay? So again, here we have the ribosome, and here we have the mRNA, all right? Simple enough. This is just right outside of the ER, okay? So once it attaches to the ribosome, what would the ribosome do? The ribosome will slightly translate it, okay? It will translate the first couple of amino acids, the first couple of peptides. And what would these peptides be? They would be the signal sequence. But again, to repeat, not to confuse you guys. What happens first is that the mRNA goes towards this area, goes towards the RER. Once it goes towards the RER, there is a ribosome that is waiting on the outside of the RER. This ribosome binds onto this mRNA, and what it does is that it translates it slightly, okay? Once it slightly translates it, it leads to there being a release or a signal sequence being produced, okay? So this signal sequence is produced with the first couple of um, mRNA that is translated for. Okay, so once the signal sequence is produced, there will be a protein called SRP that will bind onto the signal sequence. Okay, so this SRP will bind onto the signal sequence right there. Once it's bounded onto the signal sequence, it will act like a taxi. Okay, it will take this ribosome, this mRNA, towards the ER. Okay, or the ER membrane. Once it's taken it towards the ER membrane, it will bind onto its channel, the SRP channel, or the SRP receptor. Once it binds onto its channel, it will lead to energy being released, or GTP hydrolyzation. You guys just need to know that energy is being released. Once energy is released, this translocon channel kind of opens up, okay? It opens up. And once it opens up, it allows this peptide or these amino acids to kind of go into the translocon or go into the ER, all right? And once it's entered, now this SRP would kind of be released. It will go about its day. And now this ribosome would carry on translating this protein inside of the ER and kind of making it longer and longer and longer until it is fully finished. Then this translocon channel will kind of close. And this ribosome will leave. To explain it in simple terms, okay, I'll repeat one more time. There is the ribosome with the mRNA right outside of the ER, okay? Once it goes right outside of the ER, it then, once it is right outside of the ER, it will then code for the first couple of proteins or peptides. The first couple of peptides that are coded for by the ribosome of the mRNA is going to be the signal sequence. Now, this signal sequence will allow for this SRP, or the signal receptor recognition peptide, to bind onto the signal sequence. What this SRP does is that it takes it closer to the ER, acts like a taxi, and then it binds onto its receptor. The SRP will bind onto its receptor, and it will lead to energy being released. Once energy is released, it will open up this channel called the translocon. So it opens up this channel, the protein or the part of the protein will kind of enter the ER through this translocon. Once it enters, the ribosome will carry on translating the protein, the peptide, and then once it's fully developed inside of the ER, it will be released, and this ribosome will go about its day and leave the complex. Okay? Did that make sense? You guys have seemed to ask a couple questions. All that happens when mRNA is typing the code at the same time, right? The chain gets taller every. Yeah, you're correct. You're correct. So all of the time would start by having enzymes. Your question, dot, that you only have like a dot in your name. We're going to be getting into that later. I just need you guys to understand the steps of this, okay? How does the ribosome know to start at the end terminal? Wallahi, Abdullah, that is a very good question, Salah. I'm not sure if they're going to ask you a question on that though, but so that's I don't even know. But I don't think that's too important to know like how the ribosome functions and stuff. I think that's very complicated and way out of your scope. No, no. Once the SRP binds onto the channel or binds onto its receptor, it will release energy. This energy, it's used to open up this translocon channel or this gate right here. Okay. Let me just wipe it for you guys. It's used to kind of open up this translocon channel right here. Okay, it opens up. It's closed before, right? With energy, it opens it up. Once it opens up, the peptide can kind of enter. Okay, once it enters, now the ribosome will carry on translating, and the peptide will get longer and longer and longer until the peptide has fully developed, it's fully produced, the protein is fully produced. Then this translocon channel will close. Okay, once it closes, the ribosome will leave. All right. Did that make sense? Or should I repeat one more time? Okay. So here is all the steps again. If you guys would like to look over it later, be my guest. But basically what I've, explained so far. Does anyone have any questions before we move on about any of these steps? We can move on, guys. Okay, so let's move on. Now, inside of the RER, now the protein is inside of the RER, right? Inside of the RER, okay, what's up? You have a question, right? Now, I didn't mention it because it's not too important, but there is like this signal peptidase, which kind of like breaks off the signal sequence whilst it's inside of the cell. It just kind of removes it and gets rid of it. But it's not really important, in my opinion. The signal peptidase is just an enzyme present here, this one right here. It kind of just breaks off this signal protein. All good? All right. Now, for the modification of the RER, yeah. So for the modification in the RER, so once the protein is inside of the RER, it undergoes modification or can undergo some changes. Now, what can happen is that there can be protein folding that happens to the protein whilst it's inside of the RER. Can anyone remind me what proteins were responsible for this? What was responsible for protein folding? If anyone remembers, please let me know. The chaperones, thank you very much. So there are chaperones that are present in the RER, which kind of fold up the protein. What also can happen inside of the RER is that there can be N-linked glycosylation, which basically means a sugar group being added onto the RER, okay, or being added onto the protein. Sorry. Okay. Now there can also be disulfide bonds that form, which are really strong bonds. There can also be broken down or breaking down of misfolded proteins. Okay. So misfolded proteins can be broken down inside of the RER. What also can happen is that there can be transport vessels that kind of send the protein that's present in the RER to different places. Okay? So a couple modifications or a couple things that can happen to the protein whilst it is inside of its RER is protein folding, glycosylation, which is the addition of sugar, disulfide bonds, misfolded proteins being broken down, and even the transportation of proteins to different areas of the cell. Now, let's get into the different sites that the protein can go towards inside of the body. Now, we're getting into how proteins go to the lysosome, how proteins go to the nucleus, how proteins go to the mitochondria, how they get to the peroxisomes, and how they even leave the cell. Okay? So let's start with the lysosomes. Now, this picture kind of shows everything, right? All I want you guys to know is that the protein that goes towards the lysosome, always say they go through the secretory pathway. In the secretory pathway, the mRNA gets translated in and around the RER. Once it gets translated there, it can enter the RER. Now the protein that goes to the lysosome, and here it's inside of the RER. Okay? Whilst it's inside of the RER, what happens is one of the modifications that we talked about before. What happens in the RER is that this protein gets glycosylated, okay? It gets N-linked glycosylated, which basically means there is a sugar group that is added onto the protein. Now, this sugar group is called mannose, okay? It's called mannose. Not M6P, it's called mannose, okay? Once this mannose is added on to this protein is then taken in a vesicle, right? It's taken in a vesicle, and then it goes towards the Golgi. We talked about how one of the modifications in the RER is they produce these transport vesicles, which can kind of transport proteins, right, to different places. فالحين, once the mannose group has been added onto the protein, it will then go towards the Golgi. Okay? Now, what happens in the Golgi, you may be asking? Now, what happens in the Golgi is that this protein gets its mannose group, remember the sugar group that was added on in the RER, it gets phosphorylated, or phosphate group is added onto the mannose, okay? So it then produces the structure called M6P, all right? Produces the structure called M6P. Now, what this M6P will do is that it will allow for the protein to kind of enter a vessel that will take it to the lysosome, okay? And that's how it ends. What happens first is that the protein is inside of the RER. It gets a mannose group added onto it. And once this mannose group is added onto it, it is then transported to the Golgi. Now, what happens in the Golgi? This mannose group is phosphorylated by an important enzyme we'll be talking about in a bit. Once it gets phosphorylated, it will then go towards the lysosome wants to head towards the lysosomes, now it's at its destination. Okay? So again, firstly, the protein gets N-link glycosylated in the RER with mannose. Okay? Once it gets put on with mannose, this mannose allows it to go to the Golgi. Once it goes to the Golgi, there is this important enzyme that I need you guys to remember. N-acetyl glucosamine 1 phosphotransferase. Okay? This enzyme is very important. What this enzyme does is that it just phosphorylates the mannose. It puts a phosphate group onto the mannose. And what this will do is that it will attract a vessel that will take it to the lysosome. Okay? So what I will say, the protein gets glycosylated with mannose inside of the RER. It then goes to the Golgi. Inside of the Golgi, there is this enzyme called N-acetylglucosamine 1 phosphotransferase, which phosphorylates this mannose. Once it phosphorylates the mannose, this protein can then go towards the lysosome, and now we are all good. Yes, you're right. Mannose plus P is M6P. Do you guys have any questions regarding these steps and how proteins go through these stages? Okay. Do you want me to explain that one more time? Now, that is a very good question. And we're going to be getting into like how different proteins go towards different places. Oh, I think I know what you're talking about. We're going to be getting into that in the next slide. Okay, eye cell disease. We're going to be getting into that right now. But all I need you to know is these steps on what happens when a protein goes towards the lysosome in a normal person. Do you understand that? Or do you have any questions about like the process? Do you have any questions regarding the steps or can we move on? Okay, all good? All right. So, what if there is a deficiency in N-acetylglucosamine 1 phosphotransferase enzyme? Now, wow, that was a very long question. But we said that N-acetylglucosamine 1 phosphotransferase was responsible for phosphorylating the mannose group in the Golgi. Okay? It phosphorylated the mannose group in the Golgi. All right? Turning it from M only to M6P. Okay? We said that this enzyme was responsible for that. So, what can happen if there is a deficiency in this enzyme? Does anyone want to let me know? What do you guys think? Now, you guys are correct. It can lead to eye cell disease. Now, eye cell disease is a disease also known as mucolipidosis type 2, and it's caused by the deficiency of N-acetylglucosamine 1 phosphotransferase. It will lead to the things that you guys have mentioned: elevated serum lysosomal enzymes, undigested glycolipids. All these basically happen because of the proteins that are destined for the lysosomes not reaching their destination. And all this happens because of this enzyme being deficient, because N-acetylglucosamine 1 phosphotransferase is deficient and it's not doing its function. What's its function? It basically just phosphorylates the mannose group in the Golgi. Okay? And that's what happens in eye cell disease. I hope that made sense. Do I need to repeat anything, guys, or can we move on? All right. So how do proteins get into the nucleus? Now, this is a very similar process. What happens is that there is a signal, right, on the protein called a nuclear localization signal, or NLS. This signal will be bounded onto by a protein called NIR. Once this NIR bounded onto the NLS, it kind of takes it into the nucleus or through the nuclear pore. Once it's taken it through the nucleus, there is this another thing called RAN-GTP, which kind of takes the NIR back out the nucleus, and then it allows the protein to be free inside of the nucleus. To repeat one more time, there is a protein. This protein has something called an NLS, or a nuclear localization signal. This nuclear localization signal kind of causes this NIR to kind of bind onto it or attach to it. Once it attaches to it, this NIR acts like a taxi, takes it through the nucleus. Once it's taken it into the nucleus, there is this thing called RAN-GTP, which kind of attaches to the NIR and takes the NIR back outside the cell, thus releasing the protein, and now the protein can go into the nucleus and do its function. Okay? Now again, to repeat in words, what happens is that there is this protein that has NLS, nuclear localization signal. This NLS will be bound onto by a protein called NIR. This NIR will take the protein through the nuclear pore. Then this RAN-GTP would kind of bind onto the NIR and take it out of the cell. Thank you very much for that. Now, because NIR is responsible for like taking in NLS, right? But if it doesn't, leave the cell, it's not going to be able to take other proteins into the thing. Think about it like this, right? If there's a taxi that's taking people from, let's say, uni to your house, right? Now, if that taxi stays next to your house almost all the time, every single day, it's not going to be able to take other people from uni to your house. Well, that shouldn't even happen, but you guys get the point, right? So NIR is responsible for taking things into the cell. But like if it's not present outside of the cell, there's literally no point of its function. Did that make sense? Okay. So now let's move on to the mitochondria. Okay. Now, how do proteins get into the mitochondria? I know you guys probably know that the mitochondria has two layers to its outer membrane. It has the outer membrane and the inner membrane. Okay. Now, in the outer membrane, what happens first is that there is a protein, okay? And the protein has a signal. Again, it's very similar, right? There's a lot of signals. And in this case, the proteins that head towards the mitochondria, they have a signal called amphipathic alpha helix, which is at the N terminus, okay? Now, what this signal would do is that it will attract a protein called HSP70C. Now, this name looks very familiar, right? HSP70. Can anyone tell me what HSP70 was doing or what it does? We talked about it before, right? Thank you very much. It's a chaperone, okay? Now, what HSP70C will do is that it will bind onto the protein that has this amphipathic alpha helix. And once it binds onto it, it will kind of unfold the protein, okay? Now, what this unfolding the protein will do is that it will kind of help make it easier for it to kind of enter TOM, which is the channel that is present on the outer membrane. So again, the protein has a signal called amphipathic alpha helix. This amphipathic alpha helix attracts a protein called HSP70. We said HSP70 is a chaperone. Now, this chaperone, this time, will unfold the protein, okay? It will do the opposite from what we talked about. It will unfold the protein. Once it unfolds the protein, it will allow it to enter this TOM channel and go through it, okay? Now, once it's inside of this space called the intermembrane space, it will then head towards this next channel, which is called TIM. Once it's gone towards TIM, it will be met with another protein called HSP70M. And the same thing will happen. This HSP70M will kind of unfold this protein and make it more transmissible across this membrane, being TIM. All right? Now, the N terminus of a peptide, I believe, is the amine end of the peptide. We talked about it really earlier on, I think. You said that in an amino acid chain, there is the amine end, the carboxyl end. The N end is just the amine. Okay? Now, this is what happens when proteins get to the mitochondria. I hope that made sense. To repeat one more time, the protein has a signal, and this signal is called the amphipathic alpha helix, which is present at the N terminus. Now, this area will be attached to by HSP70C. Okay? HSP70, again, being a chaperone. Now, this time, chaperones, we know that they usually fold the protein, right? In this case, it will unfold the protein, okay? Sometimes it can unfold proteins. And in this case, it will unfold the protein. Once it unfolds the protein, it can then enter Tom, all right? Once it enters Tom and it's entered the inner membrane space or the intermembrane space, it will then be met with another protein called HSP70M. This HSP70M will again do the same thing, and it'll kind of unfold it one more time to allow it to enter the next channel called TIM. And now it's inside of the mitochondria. Did that make sense, guys? Any questions regarding the mitochondria? Okay. So now for the last point. How do proteins get to the peroxisomes? Yeah, you're correct, Mohammed. So how do proteins get to the peroxisomes? Now what happens, again, it's very similar. The protein has like a signal called PST1, okay? PST1. And this signal attracts a protein called PEX5, okay? Once this PEX5 is attracted to the PST1, it will take it into the peroxisome, okay? Once it takes it to the peroxisome, there will be this enzyme called ubiquitin ligase, which kind of chops off the PEX5 and takes it outside of the cell. It's very similar to the nucleus, okay? Very similar to how proteins get into the nucleus. There is the signal that is present on the protein called PST1. This signal is bounded onto by PEX5. PEX5 takes it into the cell, or into the peroxisome, sorry. Once it takes it into the peroxisome, there is another enzyme called ubiquitin ligase, which kind of breaks it off and takes it out. Does this process make sense? Now, something else that you guys also should know is that it was mentioned in your slides that a mutation of PEX2, which is one of like the PEXs, can lead to Zellweger syndrome. Now, I just want you guys to remember this point. Do you guys need to know how does it lead to Zellweger syndrome? No. Just know that PEX2 mutation can lead to Zellweger syndrome. Okay? It could ask like a small recall question on it. Now, if they say PEX5, I would say that it would not cause Zellweger syndrome. That would be a very weird question, Salaha. But it would probably, if all of the choices are very weird and the only one you recognize is Zellweger syndrome, then put Zellweger syndrome. If the answer is literally saying there will be lack of proteins getting into the peroxisome, then put that answer. Because PEX5 is responsible for like transporting proteins into the peroxisome. Okay? That's its main function. Does this make sense, guys? Yeah, they're probably going to say that. You're right, Miz. So, okay. What happens first is that there is a protein, right? The protein in this case is cargo. Now, cargo has a signal called PST1. Now, this protein with this signal, PST1, it will attract a protein called PEX5. PEX5 will bind onto it and it will take it into the peroxisome. Okay, it will act like a taxi. All right. Once it takes it into the peroxisome, there is this other enzyme called ubiquitin ligase, which kind of forces the PEX5 to kind of leave the peroxisome one more time. Okay, and leaves it, and then now the protein can stay inside of the peroxisome, and that is all that's happened. That makes sense? Okay. Now again, to put it into words, the protein has a PST1 signal peptide. PEX5 will bind onto the site. It will take it into the peroxisome. Once it takes it into the peroxisome, there's ubiquitin ligase, which kind of breaks it off and takes PEX5 out of the cell. Okay? So that's it for the peroxisomes. Now, how do proteins exit the cell? Now, some proteins are just destined for the outside, and they're released from the cell when their vesicle kind of attaches to the membrane. And there are two types of secretion a protein can have going outside the cell. There could be constitutive secretion and regulated secretion. Now, constitutive secretion is literally just the release of a protein outside of the cell without a trigger, okay? Without a trigger. It's just released out of the cell. A good example could be collagen. Whilst regulated secretion would be when proteins are like kind of stored in these vesicles, in these granules, and they're released in response to a signal. Now, a good example could be like insulin. Once you eat food, your blood sugars spike. That's when insulin's released. So proteins that are released due to a signal, they are released via regulated secretion. Whilst proteins that just are released without any trigger, they're just released kida al-fadhi. They are known as constitutive secretion. Okay? Did that make sense, guys? All right. Now this table kind of shows everything. All I want you guys to know is like the signal sequences that are associated with each protein that heads towards these areas, like PST1, amphipathic end terminal, the NLS, the M6P, the N terminal. Just know if you see one of these sequences, where are they taking the protein? Okay? So if it's the N terminal signal peptide, they're going towards the RER, right? Okay? If it's the M6P, it's going towards the lysosome. And if the M6P is not made, if the N-acetylglucosamine 1-phosphortransferase enzyme is not working, it could lead to eye cell disease. Okay? If the NLS is present on the protein, it takes it to the nucleus. If there's an amphipathic N-terminal helix, it takes it to the mitochondria. There's a PST1 that is present on the protein. It takes it to the peroxisome. Okay? And I just want you to link these three. For the ER, don't mind about it. It's not really important. And if you guys understand these concepts, then you'll be good for this lecture, inshallah. Does anyone have any questions regarding this before we move on? Now, NIR is literally just the protein that bounded onto the NLS and took it into the nucleus. It acts like a taxi, okay? The proteins that are destined for the nucleus, this NIR kind of takes it into the nucleus. And that's what NIR does. Let me show you a picture to kind of make you understand it almost. Oh, is it good? Okay. No, you don't need to know about that. Just know that it transports it into the nucleus. Okay? All right. I think we're finished the lecture. Thank you guys very much for tuning in. I know I took a lot of time. If you guys have any questions, feel free to ask it in the chat right now. If you guys have any questions after the session, here are my contact details, my email, my number. Never hesitate to ask questions. I wish you guys good luck to your midterm. Inshallah, it's very easy. And inshallah, you guys ace it. Just make sure to answer a lot of questions. Practice questions are key. Make sure to do past questions too. Do everything you can. وبالتوفيق لكل. مع السلامة. Thank you guys for attending.